Good evening, one and all. The Department of Chemistry, St. Xavier's College, Autonomous Kolkata, cordially welcomes you all to the first day of RACB'22 International Chapter 2. Your active participation and enthusiasm towards science had made Chapter 1 a grand success, and we are pleased to present to you the second chapter, comprising of engaging sessions by Dr. Saurja DasGupta and Dr. Ajay Boshak. Today, we're delighted to have among us Dr. Saurja DasGupta, whose lecture on how life was born, an RNA story, will definitely enrich our knowledge to a great extent. So, without any further delay, I would like to call upon Dr. Reena Ghosh, Associate Professor, Department of Chemistry, St. Xavier's College, Autonomous Kolkata, to introduce our speaker of the day.
Hello, yes. So I would like to thank right at the outset the head of the department, Dr. Ankur, to invite me to chair the session, because this is a session which is very important to me, because everyone knows that Saurja has been our student, and therefore it's a great pleasure and a privilege for me to introduce him. Well, good morning, Saurja. Very good morning to you. And good evening, all my dear colleagues and my dear students. So regarding Saurja's background, so everybody knows that he has graduated from Xavier's, and he has done his PhD from the University of Chicago under the supervision of Dr. Joseph, I think it is Piccirilli. It's Italian, I think, Saurja? What is
Piccirilli. Yeah.
Piccirilli, yes.
Yes.
Where he studied the structural principles underlying RNA catalysis. He is currently doing his research at Massachusetts General Hospital, Boston, as a postdoc associate, where he is exploring the biochemical milieu that could have given birth to life on Earth. Well, that is very exciting, Saurja. His primary research interests include directed evolution of catalytic RNAs or ribozymes and the development of chemical models for primitive cells. Dr. Saurja DasGupta is also a contributing editor at Club Sci-Ri, a popular science blog. Now, I think students, with whatever I have said, it's time that we now realize, I have said it time and again, that we bridge the gap between compartments that we know is chemistry. Now it really doesn't exist. And I think Saurja will, at the end of his lecture, convince you about that. I have been saying it for past how many years, I don't know, that there is nothing like organic, physical, inorganic biochemistry and nothing like that. Everything is merged and you work towards a certain goal with a consolidated knowledge. So that is my message to students. And Saurja, I think let us dedicate this lecture to the forthcoming National Science Day here on 28th February. Thank you.
Okay, so should I, can you hear me? Am I audible?
Yes, sir.
Yes, okay. And should I now share my screen?
Sure, sir.
Okay. Sure. Okay, so let me see if the full screen is visible, I'm guessing. If not, I hope you'll let me know. Okay, so thank you, ma'am, for such a, it's a great honor to be introduced by RG. It's also a wonderful honor to be invited once again by Professor Ankur. I am actually very fortunate to have been able to come back to St. Xavier's in that location and now virtually from time and again to talk to the current students in the department. You know, I listened to the talks in the last last week's webinars, world-class science, wonderful. And I'd like to take this opportunity as well to thank the organizing committee. What a wonderful job you guys have done. And the 3 years that I spent at St. Xavier's in the Department of Chemistry have had a profound impact in both my personal and professional careers and lives. So just indulge me for the next couple of slides where I get a little nostalgic. So this is a picture from, I think, the first departmental picnic that we went to. It was in Taki, I believe. This was when I learned of the many talents that my friends had outside of the classroom. The picture that you see here, the people in this picture are now spread out all over the world. I think at least in 6 different countries now that they've settled in. This is where it all began, in a way. The smells, the fumes, and the countless candid conversations. For some reason, faculty became more candid when they were in lab. You guys probably realize that as well. So this is where I work now. This is Massachusetts General Hospital, which is a hospital affiliate of the Harvard Medical School. And I'm up top there on the 7th floor. So for the next 40 or 50 minutes or so, I'm going to talk about the science that I do here. The main scientific question, as RG just mentioned, is that I'm interested in this question of how life emerged on Earth. Now, this question is a scientific question. Don't let anyone tell you otherwise. But this is also a philosophical question that humanity has been trying to answer for 5 millennia or so, without much success, unfortunately. To set the stage, scientifically at least, we can take a trip back in time, 14 billion years or so. That's when the entire universe emerges out of this weird and fantastic event that we refer to as the Big Bang. And then about 4.5 billion years ago, so all the dust particles and the gaseous particles that ultimately emerge from within the Big Bang gets pulled in close to each other by gravitational forces, and then that creates our home planet, the Earth. But at that point, Earth is a crazy place to live in. It's a newborn. It has temper tantrums. It cries a lot. So there are earthquakes, regular volcanic eruptions, meteorites that crash from outer space onto Earth. The crust starts to form. There is no proper atmosphere that is formed. So what I'm trying to say is that in this picture of the Earth, violent Earth, life is very unlikely to have emerged. And then let's take a jump cut to 3.6 billion years, which is when we have evidence for life. Now, the interesting thing here is that the life that we have evidence for, we think biochemically is quite similar to modern life, like, for example, in a bacterial cell, which means that life would not have been the very first forms of life on Earth. That life is way too complex to have emerged as the first forms of life. So that leaves us with this thin slice of about 400 million years or so when the first life, could have emerged on Earth. Now, can we discover how life emerged on Earth? We perhaps cannot discover it unless you can break the laws of physics and make a time machine and we can go back in time and observe how life emerged on Earth. There is no way that we can sort of figure out exactly, or at least tell exactly, what the chemical steps were that led to the formation of life on Earth. So it will perhaps forever remain a mystery. But not all mysteries remain unsolved, is what I'm trying to say. That's the summary of today's talk, in a sense, right? So, in the best mysteries, in the best crime thrillers, there is no witness for the crime, but somehow detectives manage to gather evidence, right? Perhaps they have a telling autopsy report, or maybe they have the murder weapon. So they use the evidence that they have, and then they make intelligent guesses, and then they reconstruct the crime scene, and then solve the mystery. Right. So what I'm trying to say here is my approach is we can look at life all around us. There is life all around us. We can take clues from the biochemistry and the chemistry of life all around us and make intelligent guesses. And perhaps we'll be able to reconstruct the origin of life in the lab. So this is what my research goals are. So the 2 topics of today's webinar, chemical sciences and biological sciences. We don't really know how chemistry smoothly transitioned and became biology, which is why the fingers in this image don't really meet. So the gap in the middle of those 2 fingers is what represents my current obsession, which is, to put it bluntly, It is that I want to create life in the lab from scratch using non-living matter that would have existed 4 billion years ago before the origin of life on Earth. Now, so some of you might think that I'm being a little fastidious here, I'm being frivolous when I'm saying that I want to make life in the lab. It's an incredibly difficult thing to do, which is why no one has done it yet. But my goal for today's talk is to convince you that it's not as hard as you might think it is. Before I do that, here's a little assignment for you, an easy question to answer. So do you think science will be able to answer how life was born on Earth? Right. So if you're absolutely sure that science will not even get close to answering this question, you can perhaps answer with a 0 or a 1 or a 2, hopefully not a 0. And if you're absolutely confident that science will be able to solve this problem sometime in the future, then you can answer with a 9 or a 10. So you can answer, if the organizers permit, you can put your numbers in the chat, if not in your head. And then I would request you once again to answer this question once I'm done with my talk. So if the number increases, then I would consider the talk to be a success. And one more thing before I launch into my thing is I will try to keep the technicalities and to a minimum and experimental data to a minimum, because I think that this is perhaps not the perfect platform for discussing a lot of experimental results. But I will try to introduce new ideas and new ways of thinking that might benefit both of us. So here goes. Those of you who answered that question with a low number, perhaps a 1 or a 2, I think one reason could be because you look at life all around you and you see that life is diverse, so many different kinds of life. And life is complex, really, really complex. Even a simple cell, like a bacteria, we consider to be a simple life form, but a bacterial cell is super complex. So you might be very easily overwhelmed that like this life is so complex. How did it even come from something simpler? What could be something that's simpler than a bacterial cell, for example? And my answer to this is that a lot of things start from 0, and then they turn out to have infinite potential, essentially. And we don't even think about those things. So one example is learning a new language, right? So when you're born, you know absolutely 0 words. But then when you're a 3-year-old, you know about 100 words, nothing to write home about. But then now as a fully functional adult, you are able to construct almost an infinite number of sentences. How did this happen? So this happened because you learned this little something called grammar when you were a kid in school, right? So grammar told you how you could take letters and words and mix them in a way that makes linguistic sense, right? Okay, so now let's take all of life that we see all around us and break it down into chemical letters and chemical words and see how we can mix them together in a way that makes chemical sense. Okay, so let's look at something that's really complex, but something that we chemists are used to looking at, a molecule. So this is Taxol. This is a natural product found in a tree. It's anti-cancerous, I believe, but that's not the point here. This is an overwhelming molecule. Okay, even chemists are overwhelmed by it. I do not know how to make this molecule. Perhaps you don't as well. Biology makes this molecule easily, but biologists cannot, so-called biologists cannot make this molecule. Why? But chemists can and have made this molecule time and again. So what do sort of traditional chemists have that traditional biologists do not have? So they have a secret tool that you all will acquire. I acquired at St. Xavier's during talks given by Dr. Ankur Rai, and I had this little red book that I used to carry with me from my second year onwards, I think. This was a book written by Stuart Warren. So those of you who don't recognize this name, you should. But those of you who do recognize this name would know what the tool is. So the tool is nothing but retrosynthetic analysis, right? So retrosynthetic analysis is basically a logical approach that chemists traditionally have used to simplify complexity. That's all there is. Now, when designing a retrosynthetic analysis pathway, there are 2 things that you need to keep in mind. One is that each step of that retrosynthetic pathway needs to make chemical sense. And then you can only stop your pathway. You start breaking down this complex molecule, make it smaller and simpler, smaller and simpler. You can only stop at a point when you reach molecules that you can just buy off the shelf from some company. So commercially available. These are the 2 constraints that we impose on our retrosynthetic analysis pathways. So a quick look at how retrosynthetic analysis works. Again, do not read into the chemistry of this because this is not an organic chemistry lecture, but you have something that's really complex, overwhelming to some, and then you can reach a point where you have molecules that look much more innocent, much more simple. So chemists have this power to do this. Now, what if you use the same kind of thinking to something that is a little more complex than a simple molecule? Let's say a bacterial cell or a simple cell. Now, a cell is actually not just a little more complex than Taxol, it's way more complex. A cell has so many different things. Eukaryotic cells have a bunch of stuff inside them. They have mitochondria, they have nucleus, they have Golgi bodies, and endoplasmic reticulum, lysosomes, a lot of different things. But when you think of a cell conceptually, I think it's fair to say that a cell is basically three things: a compartment, genes, and enzymes. So our target model of primitive cell would basically be a bag of genes and enzymes, right? I think that's fair to say. So when we embark on the retrosynthetic analysis of this living cell, each step needs to make biochemical sense, and we can only stop at a point when we reach molecules that would have been available on primitive Earth, say 4 billion years ago, before the origin of life. So I call this prebiotically available. So the prebiotic means pre-biology, as in pre-life, before life. And I will use this term a lot during my talk, so just defining it at the outset. Prebiotic means available before the origin of life. Okay, so what would this primitive cell look like? There are no physical remains, there are no fossils of primitive cells from way back when. So as I said, we look in the biology that surrounds us and try to take clues from that. Okay, so this slide summarizes almost the entirety of biology. This is called the central dogma that you might be already familiar with. So we have DNA we have genes that are made of DNA essentially, and the DNA message gets converted to RNA message. RNA is a sister molecule to DNA by these protein enzymes called polymerases. And then this RNA message is fed into this machine called the ribosome, which makes proteins, right? And these proteins are the workers of the cell. They do almost all of the work, and DNAs hold the blueprint of the cell. So they're like the chief architects in a sense. So this looks like a clean, nice workflow that nature uses for almost all life forms on Earth today. But if you look closely, you'll see there's some problems to this, right? So I just said that the ribosome can only read RNA messages. It cannot read DNA messages, which means that you need RNA to make proteins, but you need protein enzymes to take the information from DNA and convert it into RNA, which means you need RNA, or you need proteins to make RNA. So you need RNA to make proteins, you need proteins to make RNA. There's the self-referential paradox, chicken or egg paradox. What came before, RNA or proteins? One way to get out of this weird paradox is to postulate the existence of a single molecule that could function both as genes and enzymes. Then we don't have to go into the chicken or egg paradox. And the next few slides, we are going to hunt for that molecule. What could be that one polymer that could function as both genes and enzymes? And there's also a minor problem, is that proteins might be too complex to have emerged on Earth spontaneously. So what are the basic requirements of a genetic polymer? So what does a gene do essentially? A gene holds biological information. So a genetic polymer must be able to store information. There must be a mechanism in place which will help that information get copied and then ultimately propagated. Now, there is no mechanism known in biology by which a sequence of proteins information contained in a sequence of protein can be copied to make another protein sequence. It always has to go through the RNA and the ribosome. But because nucleic acids like DNA and RNA have this unique base pairing capacity, which I'll talk in detail in the next slide, it's possible to have a sequence of DNA or RNA, and then you can have that sequence be copied on a complementary sequence on top of it. And then each strand can get further copied and you can have information propagation, right? So nucleic acids like DNA and RNA can act as genetic polymers, proteins cannot. That's the message from this slide. And a quick refresher of how DNA and RNA looks. Of course, you are all familiar with this. DNA is this double-stranded duplex, double helical structure. RNA is a close relative, usually in a single-stranded form. It's a polymer made of nucleotide building blocks. So each building block is called a nucleotide. And each nucleotide further is made of three things: a nucleobase, which is an aromatic heterocycle, a ribose sugar, a 5-carbon sugar, and a phosphate, right? Now, nucleobases come in 4 varieties. So A, U, G, and C are these different kinds of nucleobases. But interestingly, the way their chemistry is structured, nucleobases can form hydrogen bonds with other nucleobases. As you can see, A can form hydrogen bonds with U, and G can form hydrogen bonds with C. And this is what we call base pairing, right? So when 2 strands of RNA that are complementary to each other come close, they would just stick to each other like Velcro, essentially. And almost all of biology, if you look at it, is put together by hydrogen bonds. Because of the hydrogen bonding capacity of RNA, intramolecular hydrogen bonds can lead to the formation of interesting structures in RNA. As you can see here, this single strand folds up into interesting structures, right? And this leads us to the next concept, which is, what are the basic requirements of an enzymatic polymer? So that for something to function as an enzyme, it needs to do a few things, right? And we all know what it needs to do. It needs to bind the substrate, it needs to catalyze a chemical reaction, it needs to release the products, and so on. But the primary condition or precondition is that this polymer needs to adopt a specific rigid 3-dimensional structure, because it is almost always that the structure dictates the function. So let's look at this 30-second clip of this RNA strand. It's a long strand of RNA, no particular structure, but you'll see how, because it can form intramolecular hydrogen bonds, it'll form this interesting sort of compact structure. So you see how this sort of folds on itself, it turns and twists because it can form hydrogen bonds. And very soon, you'll see how this long elongated string of molecule now becomes a compact structure that perhaps could be able to catalyze a chemical reaction. This looks pretty much like a protein at this point. It's all compact and rigid, right? So if you look at life around us, so we looked at the conceptual requirements of what you need to make genes, which is it needs to have information that needs to be copied and so on. And you need to have the polymer adopt a structure that is rigid. Now let's look at nature and see whether we have such examples already in nature, right? So if you look at life all around us, we know that there are creatures out there that have genes that are made of RNA, not DNA. And of course, the prime example nowadays is SARS-CoV-2, and all coronavirus classes, they have genes that are made of RNA. HIV is an example. So since RNA looks kind of like DNA, it makes sense that RNA can function as genes. But an astounding accidental discovery, actually a couple of discoveries in the 1980s, revealed that RNAs were also able to catalyze chemical reactions, which means RNAs could be enzymes. Without the involvement of any proteins, RNAs could catalyze chemical reactions. It was a shocker back then, but then it was recognized with the Nobel Prize in 1989. And then 20 years after that, the Nobel Prize was given for the discovery of the structure of the ribosome. So the ribosome is the molecule that makes proteins in our cells. So in that structure of the ribosome, it was revealed that the enzyme portion of the ribosome, the part that actually makes the proteins inside the ribosome, was entirely made of RNA. So this means that even in current biology, modern biology, you and me and everything you should see around you, all proteins are made by an RNA enzyme. So RNA makes all proteins. So this means that proteins cannot be genes, right? They can only be enzymes, but RNA can function both as genes and as enzymes. So this brings us to this very exciting hypothesis, which we call the RNA World Hypothesis, which posits that at an early stage in the Earth's history, the first forms of life used RNA as their genes, not DNA, and RNA as their enzymes. not proteins. So this also further simplifies our retrosynthetic pathway, right? So initially I was saying that a primitive cell, a model for a cell would be a bag of genes and enzymes. Since RNA can act as both genes and enzymes, then it's just a bag of RNA. So what we have to make is a bag of RNA. But before I go into how we can sort of break down a primitive cell, this is the retrosynthetic analysis that I'm proposing here. You start with a bag of RNA, essentially, which is a cell. You keep the bag aside for a second. You need enzymes. As I said, primitive life did not have proteins, but it still needed enzymes. Those enzymes were made of RNA. We call them ribozymes. But these enzymes had to be long, somewhat long, right? And had to be structured. So where did these really long pieces of RNA come from? So using retrosynthetic logic, it must have come from shorter pieces of RNA. And those shorter pieces of RNA must have come from even shorter monomeric building blocks of RNA. And we know that these are called nucleotides. And then these nucleotides would have come from even simpler molecules. So we call them feedstock molecules. And these are very simple molecules, like hydrogen cyanide, formaldehyde, carbon monoxide, ammonia, very, very simple molecules. And we have evidence for the existence of these molecules in interstellar clouds, in meteorites, on Earth. So we know that these molecules would absolutely have been present on primitive Earth before the origin of life. Okay, so before I actually start on this pathway, I could pause for a few seconds. If you have any questions, you could just ask me so that we can be all on the same page before we begin. So I can just pause for a few seconds. If you have any questions, shoot, because this is an informal discussion type session, right? If you don't, I can move ahead, and obviously we can answer questions at the end of the lecture. So going to RNA. An RNA is a polymer made of nucleotides. So, if you break a nucleotide, what do we get? We get a sugar, as I said, nucleobase, and a phosphate. Phosphate is easy. Let's keep that aside for a second. Are there chemical pathways that would have been prevalent on primitive Earth that could have created a nucleobases and sugars? So I'm just showing you two classic synthesis pathways. There are better, more efficient pathways that have been proposed more recently. Those are more complex, not required for now. So the most famous pathway for making ribose is this thing called the Formose reaction, which is, again, let's not go into the mechanism of this pathway, but what I'm trying to sort of impart, the information that I'm trying to impart here is that you take a molecule that is so simple, like formaldehyde, a single carbon molecule, and if you just leave that molecule in an alkaline pH, it condenses on itself. And after 4 condensation reactions, you get something like ribose. Obviously, you get other side products as well, but you get ribose as well in this mixture. So you start from something so simple, you get a part of the building block of RNA. Similarly, if you take hydrogen cyanide, which ironically is poisonous for life. If you take hydrogen cyanide and let it be, it polymerizes, it reacts, it condenses. It condenses on itself, and then you get to something like purine, like adenine or guanine, which are pretty complex molecules if you look at it compared to hydrogen cyanide. So you are able to take simple things that would have been present on Earth and make the building blocks, or at least parts of the building blocks of RNA. So if you have these building blocks, the nucleotides ready, you have to put them together so that they can form long or at least short pieces of RNA. But remember, at the outset, there were no enzymes, right? Enzymes are complex, they must have emerged later. So how would nucleotides come together to form long pieces of RNA? That's a major question. So I call this the assembly problem. Again, let's look at biology. How does biology make RNA? Biology makes RNA by using building blocks called nucleoside triphosphates, or NTPs. And the leaving group in this case, the reaction basically is, if you take 2 pieces, let's say you take 2 building blocks, there's a nucleophilic attack. Again, it's all organic chemistry. It's a nucleophilic attack of a hydroxyl from the first building block to the phosphate of the second building block, and then there has to be some leaving group that leaves. In biology, it's a diphosphate that's a leaving group, and the building block is a triphosphate. And you need protein enzymes to carry this reaction out. But if you don't add the protein enzyme, this reaction is very, very slow. Like it does not happen at all. So, which means that on early Earth, in early versions, when biology was being created, there were no enzymes. So there must have been some other leaving group that would have been more reactive, a better leaving group, that would have allowed for RNA assembly without enzymes. And the leaving group that we think is important in this regard is imidazole. Imidazole is a better leaving group than diphosphate. And you can see in this experiment that the gel that I'm showing you here, it's basically you take an RNA and you just add these building blocks activated with this imidazole group. And if you leave this reaction for a day or so, you will see that 7 building blocks get added by itself. No protein enzyme, no enzyme required at all. You might say this is a super slow reaction because it takes 1 day to make a +7 product. But remember, this is without enzymes. So this is definitely much better than no reaction at all. And the choice of this moiety, the 2-amino imidazole moiety you see, is not arbitrary. It was found that this molecule can be made, once again, through reaction pathways that involve molecules that would have been present on early Earth, like glycoaldehyde, and cyanamide, and ammonia, and so on. So this molecule, 2-amino imidazole, was also likely a byproduct of some of the prebiotic synthetic pathways that make the building blocks of RNA. So this leaving group is a prebiotically available leaving group. Okay, so then you have the nucleotide, which I just showed you could be prebiotically made. You have the activating group that you need for the assembly reaction to happen, also prebiotically available. But how can we put these two things together? Once again, you have molecules that are simple and prebiotically available. Acetaldehyde or any other aldehyde would do, methyl isocyanide. If you mix these things together, if you mix the aldehyde and the isocyanide and the imidazole with a nucleotide molecule, you get this activated nucleotide. And this can react. This can basically polymerize on its own to form a short piece of RNA. So that's great, right? So all these individual components that you need to create a building block for RNA assembly, they're all prebiotically available, which is great. So then what the plan of action would be, then you have these activated building blocks. They would assemble on their own without enzymes to form these short pieces of RNA. And then even slowly, these short pieces of RNA perhaps would assemble into long pieces of RNA. And when an RNA is sufficiently long, I showed you it can fold up into interesting structures and perhaps function as enzymes. And once enzymes emerge on Earth, then all these reactions will be faster because that's what enzymes do. Enzymes catalyze, or they accelerate the rates of these reactions. So there's a problem. At the outset, I told you that there are no physical remains from this lost RNA world. We don't have these physical remains. We don't have fossils of primitive life. So we are left to our intelligent guesses as to what kind of enzymes would life or primitive life have used to carry out their metabolic processes. And fortunately, there is a technique that we could use. Once you have guessed what functions would have been important for primitive life, we can actually resurrect, just like in Jurassic Park, they resurrected dinosaurs. Dinosaurs lived and died 65 million years ago. These organisms, let's say from the RNA world, lived and died 3.8 billion years ago. So way, way older than dinosaurs, right? So we can sort of resurrect these enzymes in the lab by using a technique called artificial laboratory evolution, which is what I'm going to show you in the next slide. But before that, just a quick refresher, just in case of what natural evolution is. So evolution by natural selection works like this. You have a varied population, and then you have certain selection pressure that favors certain features, certain traits, and this leads to differential survival and reproduction. So for example, in this case, you have a black rock, and there are some beetles on the rock. The dark beetles sort of camouflage, and the birds cannot see the black beetles. The birds notice the white beetles, eat them up, and then the black beetles grow old and make babies, and the next generation, we have more black beetles, right? So that's natural selection. So we can use a similar sort of thinking to design artificial evolution experiments in the lab. But the fantastic thing about artificial evolution is usually evolution works on organism level, right? As you just saw on beetles. It also works on species level. But artificial evolution in the lab can work on the molecule level, or at least in the molecule group level. So outside of cells, there are no cells involved. So I'm going to tell you exactly what we do in this process. So first of all, we make a library of RNA molecules. So we have a machine called the nucleic acid solid phase synthesizer. We make a bunch of these molecules, and each starting library contains about 10 to the 12 or 10 to the 15 molecules. So it's not a small number. It's at least 100 trillion RNA molecules in a small tube. And then we challenge this library to a particular function. Okay, do this, maybe catalyze this particular reaction. And the way we design this experiment, those sequences, those molecules in that library that can perform that designed task or designated task will be selected, and those that cannot will be discarded, right? And then those molecules that survive this selection pressure, those molecules that are selected would be amplified by a technique called reverse transcription polymerase chain reaction. I'm pretty sure all of you at this point know what RT-PCR is. And those molecules will be converted to RNA. And then again, the cycle would be repeated again and again. And after this iterative process has been performed for several rounds, if we are lucky, we get out the selected molecules that perform our desired task. We can read them individually by a machine called RNA sequencer. And this is called high throughput RNA sequencing, which is a relatively new technique that has been developed in the last decade or so. So for my purposes, the function that I want is, okay, can my RNA enzymes join short pieces of RNA to make long pieces? Because, if you are a life form based on RNA, if you use RNA for genes, RNA for enzymes, what is that one function that you desperately need? You need to make longer RNAs. So an enzyme that can do that is perhaps a really important enzyme to have in your cells. So this is the task I forced my library to do. And I went through this process for a few rounds. And then at the end of the round, end of 6 or 7 rounds, only those molecules that are able to catalyze this reaction, which is take pieces of RNA, put them together, will be sequenced and identified. And this is where I show some experimental data to show you that all of this is real. So as I said, each molecule was read individually. And the figure on the left here is, as I told you, this process is iterative, right? It goes through cycles. So the first few rounds of this evolution process, you don't see any enzymes emerging. The rates are very low, background level. But then on the 6th round, suddenly observe that there is about a 500-fold increase in reaction rate. What does that mean? It means that my pool now, because I've selected only these active enzymes, my pool is now full with enzymes. So enzymes have emerged, and then it goes on increasing. And on the right, you see a polyacrylamide gel. So for those of you who are not familiar with this, it's just a matrix that you run RNA on. It separates RNA based on their lengths. So in this reaction, what do we have? We have 2 pieces of RNA joined to form a longer piece of RNA. So the RNA that's just one piece would run faster, and the product of this reaction would run slower. Without any enzyme, you see there is no product band up here. It's all blank. But with enzyme, you can detect the product even after 5 minutes of this reaction. So these enzymes are really, really good. Okay, so now these enzymes, I told you, can join pieces of RNA together. So one can imagine a situation where this enzyme is also an RNA, right? So it has a particular sequence. So what if we break that sequence down into, say, 5 pieces, and then we try to put them together by the same enzyme? So which means that the enzyme can take small pieces of RNA and can build itself, right? So this can actually start off this autocatalytic self-sustaining cycle that is very common in life, right? But there's a problem. The problem is if you put these short pieces of RNA together without an enzyme, as I already said, the reaction is very slow. You leave it for days and nothing happens. Then we come to this really problematic point where we know that enzymes are needed to make long RNA from short pieces, but those enzymes themselves are made of RNA. So once again, there's a paradox. what, if those enzymes are made of RNA and you need enzymes to make long RNAs, then where did the first enzymes come from? That's a proper question to ask. So we were thinking long and hard about this problem, and we sort of accidentally found something that's really interesting. So what we found was that if we reacted short pieces of RNA with amino acids, these RNAs and amino acids, once again, I'm simplifying this, but these RNAs and amino acids would react to form a modified RNA species called amino acyl RNAs. Now, two things to mention here. One is that amino acids would have been abundant on primitive Earth. We already know that from very famous Miller-Urey experiment. And amino acyl RNAs are abundant, and it's everywhere in current biology, because amino acyl tRNAs are the RNAs that bring amino acids to the ribosome to make proteins, right? So amino acyl RNAs would have been abundant, we think. Now, what happens when these amino acyl RNAs are put together? When we put them together without an enzyme, surprisingly, they join up very, very easily. So if you have just RNAs, they don't link up to form a long RNA chain. But if you have amino acyl RNAs, they link up to form these long polymers that are bridged by these amino acid bridges. Very interesting sort of chimeric hybrid polymer. But what was really interesting was that these polymers could fold up into the same enzyme that makes more RNAs from short pieces. So do you see the beauty here? So you have these RNAs, you modify them with amino acids, and then you have this polymer that is a hybrid polymer that can function as an RNA enzyme and then make more of itself. So this was something that we reported this very recently, I think this month. This is a very, very exciting result that tries to resolve the problem of where the first enzymes came from. So to quickly summarize, RNA enzymes can emerge spontaneously without enzymes. These enzymes can make long RNAs faster, right? And then some of these long RNAs can behave as enzymes, which means that the self-sustaining system can emerge out of this whole process. And life, as defined by NASA, is a self-sustaining chemical system capable of Darwinian evolution. So we have sorted out the self-sustaining part to some extent. So any questions at this point? Okay, if not, I can plow straight ahead. So what did I just show you? I think I took you through this journey from really small, simple molecules that would have been present on Earth, how they combined to form the building blocks of RNA, how these building blocks could have self-assembled on their own to form short pieces of RNA, and how these short pieces of RNA then could have assembled to form RNA enzymes. And now how these RNA enzymes would have then turbocharged the whole process and made more RNA, more long RNAs faster. Now, I think the question is, we have to bring in ultimately a cell, right? Because all life as we know it is cellular. So how can we make models for primitive cells? So to reiterate, a cell to us now is a bag of genes and enzymes, so a bag of RNA. We already have an RNA, that enzyme that makes more RNA by joining short pieces. Now we have to think of what we can use to make the cell, right? The cell membrane. So again, let's look at biology. What does nature use? Nature uses phospholipids to make cell membranes. So phospholipids are made of fatty acids, two fatty acid molecules joined by a glycerol moiety and a phosphate. But once again, we think that this whole assembly process to make phospholipids would have been rather unlikely on primitive Earth, because we don't know of any pathway to make phospholipids that is outside of biology. So the sentiment that I've expressed in this talk is we have to look for alternatives that are similar to what is found in biology, but simpler, right? So what is that one thing that can behave as a phospholipid, but is simpler than a phospholipid? Now, what is a basic criteria that a membrane should have? It has to be amphiphilic, meaning that it has to have two ends. One end has to be polar, one end has to be nonpolar, like oily end. So fatty acids, which are the main components of phospholipids, have that, right? So they have a polar carboxylic acid, negatively charged polar head group, and an oily hydrocarbon tail. Now, fatty acids are very interesting because one, short chain fatty acids, like fatty acids with 12, 13 carbons, can be made naturally by natural processes, abiotically, meaning without biology. You must have heard of Fischer-Tropsch type of processes that make hydrocarbons. Fatty acids have been found in meteorites that have crashed from outer space. So there's this famous meteorite, and if you're interested, I would strongly suggest you Google the Murchison meteorite. It crashed onto a village in Australia, I believe. So this, when they analyzed this meteorite, they found a bunch of interesting things, including they found nucleobases, they found ribose sugars, and they also found fatty acids in the meteorite. So what does that tell you? That you don't need trained chemists to make these molecules. It gets made in space by itself. So we think that fatty acids could have been a good candidate to make cell membranes for primitive cells. And the main reason for that is that if you leave fatty acids, if you mix fatty acids at a certain concentration range, they will form these spherical vesicle sacs. We call them vesicles, fatty acid vesicles. And they would spontaneously assemble. Very easy to do. Anyone can do that. In fact, when you've taken a bath, you've had these soap bubbles. Those are kind of similar because soaps are also salts of fatty acids. They're not quite prebiotic, right? But those are similar. And what's interesting is that if you mix stuff with the fatty acids, suppose you take fatty acids in a tube, you mix some RNA, you shake it a little bit, leave it overnight, you'll see that those RNAs would be encapsulated within the fatty acid. So then you would have a fatty acid compartment and you would have RNAs in them. So we call them protocells, like models for primitive cells. Okay, so then how does that fit into what I'm trying to say here? So we have our RNA enzymes that join pieces of RNA together. What I did was mix the RNA enzyme, mix small pieces of RNA, mix it in the fatty acid, mix it in the tube, leave it overnight, mildly shaking overnight, and then remove all the unencapsulated pieces of RNA and the free fatty acids to isolate the protocell, right? This is a size-based separation method. And then you add magnesium to it because these enzymes cannot function without magnesium. So you add magnesium to it, and we hope that something interesting happens. But as it so happens in science, we always show our successful results, but failures happen more often than successes. So when we did that, when we added the magnesium to this tube, what we saw was that there was this precipitation. So concentrations 4 and above of magnesium, you see this cloudy precipitates forming, which means that fatty acids have a polar group, magnesium is positively charged, fatty acids at their polar head is negatively charged. They clump together and then they precipitate, which is bad because this means that the protocell that we are trying to make is now destroyed. So that's no good. But if you look at this figure, you see that at lower amounts of magnesium, it's still clear, which means that perhaps we can add low amounts of magnesium, right? That could work. But once again, disappointment. At low amounts of magnesium, we found that the enzymes did not work, right? In this plot, you see how at 2 millimolar magnesium, even less than 2% reaction happens at 2 hours. That's miserable, very disappointing. So there are two ways to solve this problem. And this is not just observed by us, it has been observed in other papers as well. It's a recurring problem in this field. So two ways to solve this problem. One, can we identify more robust molecules to make our cell membranes instead of fatty acids? That problem is hard to solve. And the other option is, can we make these RNA enzymes function? Can we force these RNA enzymes to function at low amounts of magnesium? So that seems to be more doable. At least that was how it seemed to me at that point. So one way to do this is to go back to the drawing board, go back to our artificial evolution experiment, and then force these enzymes to be selected at low amounts of magnesium, right? And I did that, I'm just showing one piece of data. This is a paper that we are about to submit very soon. This is where we show that you can, in fact, evolve RNA enzymes that can function at low magnesium concentration. So you see here, while the old enzymes don't work at all, The new enzyme in orange shows about 30% yield at 2 hours. That's great. But during that time I was working on this problem, I also found something that I think is more interesting, especially from a chemist's standpoint. I found that to the old enzymes that don't work at low magnesium, if you add a pinch of small molecules like ribose or ethylene glycol, these enzymes magically get activated at low concentrations of magnesium. So even at 1 millimolar, it did not work, right? Without anything, it did not really work. But now with these molecules, it suddenly shoots up and works. And interestingly, as I said, ribose, ethylene glycol are prebiotic molecules. I already told you ribose has been found in meteorites. Ethylene glycol has been found in interstellar clouds in space. So it's amazing that these molecules present in this reaction mixture can enhance the reaction so much. So now we have two very different but useful ways in which you can force these enzymes to work at low magnesium. And what's good about this is now that the fatty acid vesicles would not be disrupted at low magnesium. So this is what we did. Once again, to reiterate, we took the fatty acids, added the RNA enzyme, added our substrates, mixed them, separated the encapsulated RNA in the vesicles by size exclusion, and then added the magnesium. And this time we were very nervous. Would it work after all? And then after waiting for a couple of hours, we run this gel once again that separates RNA according to their sizes. So once again, the reactant is this one single piece. It will run faster. The product will run a little slower. And even after 1 hour, we see the product has been formed. So we were really happy. And if you quantify the yields, if you quantify these band intensities, you will see that the enzymes do almost as well inside of these protocellular vesicles as well as outside. So these enzymes can function inside model protocells. And the function that it performs is to make more RNA from short pieces, which is wonderful. So if indeed primitive cells had membranes made of fatty acids, then this was perhaps the first time in 4 billion years that an RNA enzyme made long RNA from short pieces inside cell-like systems. So that was very, very exciting for us. Now, what happens when a primitive cell starts to make a lot of RNA? It grows fat due to osmotic pressure, and then it would perhaps grow and then divide. And that's where we want to go. But I'll show you a quick video of an empty fatty acid cell, protocell, and what happens when we add extra fatty acid to that protocell. So this is a spherical vesicle, a spherical fatty acid protocell, and see what happens when you just add some fatty acids. You don't need to do anything, just add some fatty acids to it. And this is an image under a microscope. So you see how when you add fatty acid, it becomes long and stringy, and then by a process called purling, it breaks apart into multiple small vesicles. And this is kind of reminiscent of how cell division happens. But this is empty fatty acid vesicles, right? So the point that I'm trying to make here is dividing compartments, dividing cells is not that hard in a prebiotic sense. What is hard is replicating the genome and the enzyme. So when a cell divides, the cell membrane divides as well in biology, but the DNA also replicates, right? So each cell has a part of its genome. But developing the self-replicating RNA is extremely, extremely difficult, and no one has been able to report a perfectly self-replicating RNA yet. If we can do that, then I'll show you what amazing things can happen. But as I said, it's not yet done yet, but that's one of my goals, to use artificial evolution to artificially evolve such self-replicating RNAs in the lab. So what will the self-replicating RNA do? It's an RNA enzyme that will take building blocks and make copies of itself, and we'll see exponential growth of these enzymes. So 1 to 2 to 4, and so on. So before I show you what happens when we manage to get these self-replicating RNA, this is a microscopy image of how these fatty acid vesicles look like. So the fatty acid membranes have been labeled with a red dye, a rhodamine dye that likes to bind to fatty acids, essentially. So you see the red color outside, and the RNA is labeled with a green dye. So you see it's green inside. So these are how fatty acid vesicles and protocells look under the microscope. Now I will show you an animation. So this is not experimental data, I have to say upfront, but this is what we expect to happen when we have a truly self-replicating cell. Right. Okay, so this is what happens. So we have these building blocks outside. You have the fatty acid compartment. Let's see what happens. So the building blocks would permeate, diffuse inside your fatty acid protocell. And then the enzyme right here would take these building blocks and make copies of itself, right? You see it's making copies of itself. And then when the cell slowly starts to divide, each daughter protocell will keep one copy of that enzyme, right? And when it splits and becomes two, each daughter protocell now has one copy of that enzyme. There you go. And if this happens, then this thing, if this happens, really has profound implications. So imagine you have two protocells with slightly different enzymes, because you know that copying has errors, right? So you copy a molecule, it will generate a slightly different molecule. So let's say in two protocells, one of them, this one has a slightly better enzyme, so it can make RNA faster, right? Which means that it can grow faster and make babies faster, which means that this lineage of this kind of protocells will quickly take over the entire population. What does that mean? This is where Darwinian evolution has spontaneously emerged out of a purely chemical system. Nothing was biological here. It was all chemistry that we put together in a certain way that you can perhaps have Darwinian evolution emerge from this purely chemical system. And once again, if you look at the definition of life, self-sustaining chemical system capable of Darwinian evolution. So which means that at this point, we can say that life is born once again. But of course, we are yet to observe this in the lab, which is why we need more ambitious and young minds to work on this problem and join those of us who are trying to create life in the lab. But I think I was able to at least show you that although we are not there yet, we are tantalizingly close to ultimately making a life in the lab and observing Darwinian evolution emerge in purely chemical systems. What an amazing feeling that would be to see something like this. In the meantime, we are looking for life outside our home planet, Earth. So this is the Mars rover called Perseverance, and it's actually scouring the surface of Mars looking for signatures of life. One of our collaborators has sent a sequencer, a nucleic acid sequencer that is looking for things like that might remotely resemble DNA or RNA. I'm not very optimistic that we find anything, but it's good to think about. And this field of origin of life research is now known as astrobiology. Is actually not a field. As RG said, it's a mishmash of all different fields of science. And this is a perfect example where boundaries are all blurred, because you have geochemists trying to figure out what happened on early Earth. You have astrophysicists, astrochemists, organic chemists, biologists, microbiologists, all coming together, sharing their perspectives, because this is just such a big problem. A single field it's impossible for a single field to come with an answer. So we have to sort of have a confluence of different perspectives. And the questions are big questions, right? This is such a fledgling field that all of the questions are unanswered. So what I'm trying to say here is that if you're interested in this sort of thing, this is the perfect time to come on board, because you have really big questions to answer. All of those questions are unanswered. So are we alone in this universe? The biggest question. And then, of course, what other planets can support life? Now we are trying to look for exoplanets that can perhaps support life. Is alien life made of the same stuff? Like, do they have DNA, RNA, proteins, similar material? Another question that you can ask is, does life need matter at all? Can information be matter? We know that information can be propagated, it can be replicated, and some variations of information can be perhaps propagated faster, fake news, especially in the age of social media. So do we even need matter to constitute life? Which brings us to this big question of, can we redefine life? Do we need a new definition for life? And these are single questions, but they actually contain hundreds of questions within them, which you perhaps can help answer. So once again, I asked you a simpler question. Do you think science is able to answer, or will be able to answer, how life was born on Earth? So once again, please share what you think about this. give an answer, give a number. Hopefully the number increases after this talk. I would quickly like to thank the group that I work in. Stephanie Zhang and Alex Radakovich, the two PhD students that I've extensively collaborated with for some of the work that I've shown you here, and Jack Shostak, who has provided me the space and the resources to carry out this exciting research. I'd like to thank him as well, and some of the funding agencies like HHMI, NASA, and National Science Foundation for giving the money to do this research. I thank all of you for giving your time and listening to what I had to say. I hope that I was able to at least share a fraction of the excitement that I feel for this field. I'd like to thank this college for all that it has given me over the years. As a senior, I think I am allowed to give you some free advice. I will just give you one piece of advice, which is this. So for those of you who want to get into academic research, I mean, I'm sure you know that it's not easy and it's sometimes not very rewarding. The rewards come few and far between. But if you still decide to get into academic research, I think it's very important to choose the problem that you work on, because you're going to give the same amount of time, the same amount of effort. So why not work on a problem that is significant? And I have 3 criteria for choosing a problem. It might work for you. It has worked for me. One is that you must be absolutely interested in the problem, not just that the problem is easy. You think that the problem can be solved fairly easily during your PhD period, but the problem needs to be really, really interesting. And the second thing that I think, which follows from the first point, is you really should want the problem to be solved. It doesn't matter who solves it. If your friend comes out and solves the problem, you should be really excited. For example, if someone else comes and makes life in the lab and I cannot, I would be really happy that this could be done. And the third point is that you should be excited to talk to people about what you do in the lab, right? Your family, your friends, your scientist friends, your non-scientist friends, and when the time comes, your juniors. So you should be able to really be excited when you talk about your research. So if your problem satisfies all these 3 conditions, then I think you should absolutely go for it. So that's all I had for you today. Hopefully you have interesting questions for me. Thanks.
Saurja, I would say just one thing after your talk, that is, you are a dreamer, and it is the dreamers who make science today and the scientists of today. And you are our reward. Okay, so you started with something very simple, but the moment you talked about the RNA World Hypothesis, many of us, we realized that, how difficult a project you have landed yourself in, because many things are yet to be proved, and you're on your journey, Saurja. So please do continue with it, and we would be extremely happy to hear about the results that you have. And the point which I was asking AR right at the beginning, after I came to know about your topic, that is the semester 6 students, if they were in the same virtual platform, I think they would have bombarded you with questions, because in semester 5 they go through this whole exercise of the basics of biomolecules, but unfortunately they are not on this platform. Anyway, I sincerely think they are very much excited and they have got ideas, and I would like them to be dreamers like you. And that is, what we, that is the effort that we, try to impart to our students. So Saurja, just thank you. I think thank you is not enough. It was really exciting.
Thank you. I mean, for those who could not be on this platform because of size issues, you have my email. Please, I would be very happy to answer scientific, non-scientific questions. Just shoot me an email. I will get back to you, I promise. And anyone with any questions at all? Was it too easy to understand, or was it so hard that there is no question?
No, sir. we have some questions from the YouTube chat. So
Okay.
first of all, I would like to thank you for taking out time from your busy schedule and sharing such an enriching lecture with us. I hope this session has enlightened our audience on this topic as well. So the first question which we have is, why enzymes cannot function without the addition of magnesium?
Wonderful. Okay. So should I be able to see the questions in the chat, or can only you see the questions?
No, actually,
Because
it's not given in the chat.
Okay, it's in somewhere else.
Forward.
Okay, so the question was that why is magnesium needed for the RNA enzyme to function? This is actually a very important question, and there are two reasons for it. RNA is, as you know, is a polyanionic polymer, right? So it has phosphates in the backbone, which means that when it tries to fold into an enzymatic or catalytic structure, there's a lot of charge repulsion in the backbone. So magnesium sort of sits between and neutralizes the charge and helps the RNA to fold, which means then it can function. So one function of magnesium is that it helps the RNA to fold into its enzymatic structure. And in some enzymes, we know that magnesium also plays a direct catalytic role. So we have things called metal enzymes, where some enzymes use metal ions as cofactors. So in some RNA enzymes, these magnesium ions go into the active site and help stabilize the transition state, for example. So there are two reasons.
Okay, sir. Sir, the next question which we have for today is...
Can I request you to not call me sir, please? Saurja is fine.
Okay, so the next question is, how can RNA structure act as genes for carrying genomic information?
So again, we already know that there are viruses that have genes made of RNA. And as I showed you, to function as a gene, it just needs to have information, and the sequence of RNA, the sequence in which the AU, GC nucleotides are placed, holds the information, and then that information can be replicated by base pairing, which means that then genetic information can be stored, it can be replicated, it can be propagated. That's all you need for genes to function. And in case of viruses, right, there is a genome that is made entirely of RNA that gets directly translated by the ribosome to form proteins. So that's how viruses with RNA genes work. I hope that answered your question. Right. You need information transfer. That's, I guess, the basic principle here.
Okay. So following this question, the third question which we have for today is, based on what criteria we can say that RNA enzyme is, or a weaker enzyme is better? I'm sorry.
Could you repeat the question, please?
Yes, it's my fault. The question is, based on what criteria we can say one is a better RNA enzyme or it's a weaker one?
Yeah, it's just like in any catalyst. So the job of an enzyme is to make the reaction faster, right? So the enzyme that makes, so in this case, this was basically take building blocks and join them together to form long RNAs. So the enzyme that does it faster, has higher reaction rates, is a better enzyme. An enzyme that does it slowly is a worse enzyme. And why an enzyme is faster and why an enzyme is slower, it's very difficult to say that by looking at the enzyme sequence. You have to do some further mechanistic analysis to do that. It's very difficult to say. But the criteria is, again, reaction rates, essentially, chemical kinetics. That's all.
Okay, so the next question which we have for today is, are protocells similar to co-acervates? I hope I'm pronouncing it right.
So I think they mean coacervates. Very interesting question. So coacervates, I'm very happy that you know about coacervates. So coacervates are a hot topic in the origin of life field. So coacervates are basically membrane-less compartments. So they're like oil droplets, essentially. So if you mix, let's say, a lot of peptides together, right? And then they found these things called coacervates. The difference between protocells that I showed you and coacervates are that protocells have fatty acid membranes, right, that allow certain things to come through. So in a fatty acid membrane protocell, small things can go in and out, but large RNAs like RNA enzymes cannot go outside, which is good because we don't want to lose our enzymes. But in a co-survey, everything can go in, everything can go out because it's like an oil droplet. Which is why I personally, and a lot of people in the field, don't think that co-surveys could have been primitive cells, because everything would come and go, so there would be no real benefit for a particular function inside of a co-survey. So in normal cells, we have semi-permeable membranes, right? Certain things can go in, certain things can go out. So I think we need that regulation in a primitive cell. So they're different in the sense that co-surveys don't have membranes. Protocells that I showed you have membranes.
All right. Okay. So the next thing which I am very much eager to know is that based on the current study of RNA synthesis, like what future can we expect from this?
RNA synthesis. Okay, so so I presume you are talking about how RNA is made in the lab. Is that what you're talking about?
Yeah.
Yeah. So the one huge sort of result for efficient RNA synthesis in the lab is the Pfizer and the Moderna COVID vaccines, because we know that those vaccines have mRNA in them, right? And to make these really long mRNAs, you need really good ways in which you can synthesize RNA. So there are two ways that you can make RNA. One is by using this synthesizer machine, and the other one is through enzymes, where you take DNA and then you add this enzyme called T7 polymerase, and the enzyme makes long RNAs for you. Now, if you can make really long RNA enzymatically, it's wonderful, because now I think there's a malaria vaccine in the pipeline from Moderna, and there are other mRNA vaccines that are in the pipeline. So if you can make really long RNAs, you can make really good mRNA vaccines. That's the therapeutic side. Also, like if you were there for Devlina's talk last week, she was talking about how you can use DNA to make nanomaterials. You can also use RNA to make nanomaterials. That field is a little less developed because RNA is a little more unstable than DNA. But you can envision scenarios where you can make a nano cage or something like that with really long RNAs that you make in the lab, and you hide a drug inside, and then you put this inside the body, and then the nano cage sort of breaks down and the drug is delivered. So you can have many applications for really long RNAs. Yeah.
Thank you. Okay, so we have another question as well. It's about the Hachimoji DNA. So the question is like this: is it possible for these RNA enzymes to join together nucleotides for DNA synthesis and replication apart from the normal RNA?
Wow, awesome. This is a great question. And I was actually associated with the paper that reported the Hachimoji sequence. But that's actually a very, very good question. And the ones that it's all about how you evolve the RNA. So the RNA enzymes that I evolved use just the standard 4 letters, A, U, G, C. But if you design the experiment in such a way that the selected enzymes can only put together building blocks that are made of the Hachimoji sequences, then I think you will get enzymes that can join Hachimoji sequences together. But the only problem is that since, as I showed you, the way the protocol works, you need things like RT-PCR, like reverse transcription, polymerase chain reaction. The enzymes that we use to do those things work usually on normal DNA and RNA, because those are obtained from normal biology. So what we are doing, not our lab, but some other labs, are trying to evolve new protein enzymes that can do PCR, for example, on Hachimoji sequences. And if you can do that, then that's great. Then you can go through the entire selection process and select. You can even make RNA enzymes that themselves contain Hachimoji nucleotides. That would be great. And we have actually made not enzymes, but there was another paper that I reported earlier. It was an aptamer, which is something that it's an RNA molecule that binds to something, right? A protein. That aptamer contained 5 letters. So A, U, G, C, and there was another new letter called P, which was part of the Hachimoji framework. You can do that if you want to, yes.
All right. So following this question, we have another question as well, which is, can it be assumed that the activation energy required for the first molecule to form was provided by lightning?
First molecule to form. Lightning, that's actually a very nice question, but lightning, I'm not sure, but there has been a lot of talk about how UV radiation, because the atmosphere was not fully formed yet. The ozone layer was, say, not formed. So UV very easily came and fell on Earth, UV light. And we have some evidence to show that UV light could have made a lot of these synthetic pathways that I showed you to make nucleotides much easier. So UV absolutely played very, very important roles in making the first molecules. Lightning, I'm not so sure, because lightning might be a little too harsh. Perhaps it might just destroy everything. Yeah. But, in the Miller-Urey experiment, they used electric discharges to make amino acids. So I'm not sure it would work with RNA, but it might again work. So you have to try to recreate these scenarios in the lab and see what you get. A lot of times it's like you cannot really plan what you do. You just try things out and things pop out that make sense. And then you go back and try to figure it out.
Okay, so we also have a question asked by Professor Indranil Chakraborty, IC sir. So the question goes like this: how do you assign a job to your library in this wonderful experiment?
Oh, thank you for calling it wonderful. So the job, so as I said, you have a library and then you challenge it to do a certain task, right? So that task could be catalyzing chemical reaction, binding to some protein or some small molecule. So in this case, I didn't want to go into the details of the technicalities of this process. But one way that you can sort of select enzymes that join two pieces of RNA is by this. I hope I will make sense. So you have the enzyme, you have the library molecules, you attach, so you need two pieces that needs to be joined together, right? So the first piece you attach to each molecule in the library through some linker. And the second piece, you add a biotin tag to it. I'm not sure if you're familiar with biotin, most of the students here, but you can add a biotin tag to the second piece. And when this enzyme can catalyze the chemical reaction, what happens is that that second piece of RNA with the biotin gets joined to the entire library sequence. And then you can use something called streptavidin, which forms really strong interaction with the biotin. I can add the streptavidin, the biotin tag, second piece of RNA, which is already now connected to the entire library sequence, get pulled down by the streptavidin, and then you can separate it. I'm sure I didn't make a lot of sense, but I'm very happy to explain this to IC personally. And you can read the paper. By the way, the paper is already out. We published this paper 2 years ago. So if you're interested, you should look up the paper.
All right. So with this question, we come to the end of the question answer session of today's webinar. Thank you, sir, for patiently answering to all the queries of our audience. Now, I would like to call upon our professor, Dr. Rahul Sharma, Assistant Professor, Department of Chemistry, St. Xavier's College, Autonomous, Kolkata, to deliver the official vote of thanks.
It is a matter of great pleasure for me today that I was extended this privilege to deliver this vote of thanks on this occasion. Firstly, from our side, we are immensely grateful to Dr. Saurja DasGupta for taking time out from his rather busy schedule and delivering such informative talk for all of us to enjoy. I'm sure your work, your words of inspiration will have a strong impact on our audience, and it will motivate all of us to rather work harder in the field of science and research. Now, exactly to the week, I think, maybe 2 years back, Dr. DasGupta was with us at St. Xavier's College, and most probably he delivered a DBT-sponsored lecture, an offline lecture. So I hope we can invite him again pretty soon. I would like to thank the audience for their cooperation and enthusiasm to make this whole program a great success. I think we had more than if I'm not wrong, and if I can quote the number, we had more than 250 people, isn't it? I hope so. Maybe more than that. So it's not like there were people only present in the Zoom lecture itself. There were people present in the live YouTube lecture too. So, and most of the questions were from the YouTube section. So thank you for your cooperation. And of course, any kind of an event is not possible in our institution without the full support of our respected Father Principal, Vice Principal, and the guidance of our other professors. I would like to thank Dr. Reena Ghosh for the department for all his help on organizing the whole seminar. And lastly, I would like to thank the organizing committee and our students who worked really hard, showed their utmost dedication so that this whole process was as smooth as we saw it. So thank you. Thanks a lot.
Thank you.
With these words, we come to the end of day 1 of chapter 2. Hope all of you have enjoyed the session. And now I would like to request all the participants to fill up the feedback form rolled out in the YouTube live chat within the stipulated time. We will be back again tomorrow with another amazing session by Dr. Ajay Boshak. So don't forget to join us tomorrow at 5 p.m. sharp. Thank you all and good night.